Okay. So so the inhibitor can either bind to the active site or into the allosteric site. Again, so if the inhibitor binds to the to the active site, we call it as competitive inhibitors because the inhibitor is competing with the actual substrate to bind to the active site. For that type of inhibition, how to overcome? You have to increase the amount of substrate in the reaction. So when you increase the amount of substrate into the reaction, there is a higher chance of substrate binding to the active site instead of the inhibitor. And then for the other one is non-competitive inhibitor where the inhibitor binds to the allosteric site. Okay, so non-competitive because the inhibitor binds straight away to the allosteric site and causing the enzyme to change shape. It doesn't care about the substrate. It just goes straight into the allosteric site, causing the enzyme to change shape. Active site changes shape. Substrate cannot enter. Reaction cannot occur. Okay. So for that, it produces, for these types of inhibition, it produces, as you can see, it has three types of graph. So the green graph shows you the normal rate of reactions, without any inhibitions. So rate of reaction occurs until reaches maximum velocity, maximum rate of reaction, where it reaches the constant rate of reaction, as you can see from the graph. So constant rate of reaction indicating that the enzyme is saturated with the substrate. So but for this one, the blue graph here, so it is the reaction that has competitive inhibitor. So as you increase substrate concentration, the rate of reaction will increase and complete ataupun reach constant rate of reactions because there's higher chances of substrate binding to the active site. The reaction also reach maximum rate of reaction or constant rate of reaction. But for the red graph here is because due to the presence of non-competitive inhibitor, so where if you keep on adding substrate, it does not influence the rate of reactions. The inhibitor will just bind to the allosteric site, causing enzyme cannot function. Okay, enzyme changes shape, substrate cannot bind. Okay. So maybe one of the way of you to overcome the inhibition is for you to remove the inhibitor. Okay. Right. So then we have reversible or irreversible. Okay, reversible or irreversible. Sorry, irreversible or reversible inhibitor. So irreversible means that once the inhibitor binds to the active site, for example, so it binds permanently and causing damage to the enzyme. Enzyme cannot function altogether. It destroys the enzyme. Okay? So the effects is permanent. Okay, permanent. And then for the reversible yang tadi lah, reversible, it falls into two types, competitive or non-competitive. As long as the inhibitor binds to the active site, it causes the enzyme activity to be inhibited. Okay, so buatlah satu mind map kan, so you have inhibition, enzyme inhibition, so bahagi kepada dua, reversible, irreversible. Okay, so reversible, it has competitive, non-competitive. So for the irreversible, you just list out all its character. Binds tightly, permanently, causing damage to the enzymes. So these are all the types of inhibitors. You have the cyanide, for example, and then cyanide, and then serine, so which is gas, binds to the enzyme acetylcholinesterase, causing neurotransmitter cannot be removed from the synaptic cleft and causing continuous transmission of impulse. So causing continuous contraction of muscle and then causing lastly death to that person. Okay. So cyanide is from smoke from fire, domestic fire. Okay? So I've gone through this on our last lecture. So this is just to recap. So so now for our lecture for today, we are going to go to the next subtopic, which is regulation of enzyme activity. Okay, so we are going to look at what are the conditions within the cell that regulates the enzyme activity inside cells. Okay, so and then we are going to look at some mechanisms. Okay, so some mechanisms such as, for example, feedback inhibitions, allosteric regulations, and then we have cooperativity. So those falls under regulation of enzyme activity. I repeat, allosteric regulations, and then we have feedback inhibitions, and then we also have cooperativity. Okay, so those are the different types of mechanism of enzyme action that falls under this subtopic. And then we are also going to look at um, the different classes of enzymes. Can so you can classify enzyme based on the types of reaction it catalyzes. And then the last one we are going to look at uh, the applications of enzyme, which we are going to look at the concept of immobilized enzyme, okay, which is enzyme that we attach to some support material and then to catalyze the enzyme reaction. Okay, so that is the application which is applied mostly in the industry, can to produce a lot of products. Okay, so let's look at this subtopic first, which is 4.7, regulation of enzyme activity. So regulation of enzyme activity is important to coordinate numerous metabolic processes. Okay, so cell can regulate its metabolic pathway by controlling when and where its various enzymes are active, by switching on and off the genes that encode specific enzyme. If all metabolic pathway operate simultaneously, there would be chemical chaos. Okay, our body can the uh, organisms basically are made up of cells. Okay, so in order for organism to functions, so the cells of that organism, it has to undergo many metabolic reaction inside cell. So these metabolic reactions, metabolic processes, all the chemical pathway that occurs inside cell, it is catalyzed by many enzymes. Okay, by many enzymes. So enzymes, we know that it is protein. Kan? It is protein. So inside our cell, we know already, inside our cell, there's the nucleus. Inside the nucleus, there's the DNA. Kan? So DNA here. Okay, so the DNA. So DNA, it has many genes. Kan? Semua sel dalam badan kita ni, composition of DNA is the same. Be it the cell in your cheek, the cell on your hands, kan, the cell that makes up of the whole body contains the same DNA, the same DNA composition. The DNA of your the cells that make up your pancreas, the cell that makes up your liver, the cell that makes up your gonads, for example, whatever cells within the body, it has the same DNA, the same DNA with the same genes. But so not all the genes in your DNA will be expressed. Means that not all the genes will be used to synthesize the proteins. Okay, so the protein. So for example, the cells of your cheek also contains genes that code for the synthesis of insulin. For example, insulin is hormone, can hormone. So hormone made up of protein. So does your cheek cell produces insulin? Insulin? No, only the cells of the pancreas produces insulin. Okay, so each cell will regulate its gene expression through the process of transcription and also translation, where the genes will be switched on or off. If the is switched on, for example, the cell of the pancreas that has the gene for insulin, for example, so the cell of the pancreas, when the gene for insulin is switched on, so the insulin can be synthesized, kan? But the gene of the that code for the synthesis of insulin will be switched off in your cheek cell. For example, your cheek cell cannot produce insulin. Macam tu lah, okay? So the DNA within your cell in all your body has the same genetic composition, but only certain DNA, certain genes will be activated at certain cells. Okay, so not all. So the DNA it has genes that code for the synthesis of specific protein. DNA has the information, okay, the information because DNA is a large molecule. So that certain region that has the gene will be used to synthesize mRNA. mRNA is a shorter molecule that copies the information from DNA. So mRNA copies the information from DNA through the process of transcription. It has the mRNA that has the information to synthesize, for example, insulin. So the mRNA is synthesized in the nucleus. It then travel into the cytoplasm, where the process of translation occurs. So translation, you have what? Inside the cytoplasm, there's the ribosome. Okay, so the ribosome is the organelle that synthesizes the protein. So ribosome, it has large subunit, small subunit. The mRNA will go in between, and then the ribosome will read the information, the code on the mRNA to synthesize the protein. Okay, so the protein. So if it's the pancreas, it produces the hormone insulin, for example. So or different types of enzyme in different types of organ, or different types of protein in different types of cell. Okay, so so all your cells will not activate all the genes. Okay, so if all the genes are activated, it will create chemical chaos. Okay, so only certain genes will be activated at certain cells to produce certain proteins, certain products. Okay, boleh faham? Okay, alright. So itulah dia. So kita pause kejap, ada azan. Okay, so let's continue. Okay, so we know now, okay, inside our cell, there's the DNA that has the information to synthesize protein. So the protein, in this case, is the enzyme that we want. Okay, enzyme. Okay, so it says here, how does regulation of enzyme activity helps control metabolism? Okay, how does regulation of enzyme activity controls metabolism? So basically, how does the enzyme inside your cells is controlled to catalyze certain activity inside cell? Okay, so for example, we have the first one. We have here inactive enzymes such as pepsinogen. Okay, so for this one here, the enzyme here is pepsinogen. So pepsinogen is the inactive form of enzymes. Okay, so pepsinogen is inactive form of enzyme pepsin. So we know that pepsin is the enzyme that is used to digest protein in your stomach. Okay, so your stomach will have cells that produces the enzyme pepsinogen. When the enzyme is secreted from the cell lining your stomach, it is released into your stomach cavity to digest protein. But how to activate the pepsinogen? So in order to activate the pepsinogen, there's that's why your stomach produces hydrochloric acid. Okay, hydrochloric acid. Apart from the hydrochloric acid is to kill off bacteria that enters with the food that you eat that may be contaminated. Another function of the hydrochloric acid is to activate the pepsinogen. So the pepsinogen, due to the acidic condition, it by the hydrochloric acid, by the presence of hydrochloric acids, it activates the pepsinogen to become pepsin. Then only the pepsin can digest protein in the stomach. Okay, so that is how pepsin is regulated due to the acidic environment. Nampak tak tajuk dia? Regulation of enzyme activity. The first example, the environment must be in the right conditions. Kan? So in this case, the pH has to be in the correct pH, the optimum pH. In this case, pepsin has to function in acidic condition. That is one form of regulating the enzyme activity. Okay, so second, presence of inhibitor. Okay, presence of inhibitors such as reversible inhibitors. Kan? So for the reaction to occur, the enzyme has active site, while the active site substrate will bind, and then reaction occurs to produce product. Okay, so but you do not want the product to continuously produce and causing an excessive amount of product to be produced inside cell. So there may be inhibitor that is present to inhibit the reaction. Okay, presence of inhibitor. So presence of inhibitors, you do not want your cell to produce excessive amount of the product, macam tu kan. So that is the use of the inhibitor. So inhibitor is not so bad, bukan maksudnya jumpa inhibitor, tak apa, dia akan stop activity of enzyme and then it gives you a negative effect to the cell. No, it does have positive effects kan. So to stop cells from producing excessive products. Okay, so that is one of the example of the positive effects of the inhibitors kan. So presence of inhibitor will also control the enzyme activity, regulate enzyme activity. Okay. So and then we have regulator molecule. Okay, so regulator molecule may activate or inhibit enzymes such as allosteric regulators. Okay, so normally we know that one enzyme, it has one active site. Okay, there are enzymes that have several subunits that have several active site, and also when the subunits are attached, it forms the allosteric site. An allosteric site. For example, here, if I were to draw, okay, so normal enzyme, it has active site. So the active site is here where substrate bind. Other enzyme, it may consist of several subunits. For example, here, and one subunit, and then we have here one subunit, and then another subunit, and then also another subunit. Okay, so this enzyme consists of several subunits where it has several active sites, and where the subunit meets, it produces the allosteric site. Okay, allosteric site. So allosteric site. So allosteric site is where regulator molecule will bind. Regulatory molecule binds can. So regulatory molecule can be uh, activator to activate the enzyme, or it can be inhibitor to inhibit the enzyme. So if the activator binds to the allosteric site, it causes all the subunit to be activated. Okay, all the subunit to be activated. But if the inhibitor binds to the allosteric site, it causes the enzyme to change shape, and all the subunit will be inactivate, and enzyme reaction cannot occur. So that is allosteric regulator. It can involve activator, allosteric activator, or allosteric inhibitors to bind to one of the allosteric sites. Okay. So for this one, we are going to look at later the concept of feedback inhibitions. So, yeah, feedback inhibitions. So that one we are going to look at later. Okay. So that involves allosteric regulators. And then sama juga feedback inhibitions, but the konsep yang sama. Okay. So the end products there. Okay, so the end product may inhibit enzyme where it involves feedback inhibitions. So you have the initial substrate, okay, binding to the first enzyme, and then you have a series of reaction occurring, and that involves several other enzymes, several formation of intermediate products, and at the end it produces the end products. So after the end product has been produced at certain concentration, can a certain amount that the cell requires. So the end product here, the production of the end product will be stopped for a while to allow the cell to use that product. Okay, so the end product will then, how to stop the reaction? The end product will bind to the first enzyme of the pathway by binding to the allosteric site of the first enzyme and stops the reaction for a while to allow the cell to use that product. Okay, so that is feedback inhibition. So we are going to look at the explanation later. Okay. So these are some of the conditions where enzyme activity are regulated. So the first one requires certain optimum pH, or even a certain optimum temperature, and then there's the presence of inhibitors, and then there's the presence of allosteric regulators, and then also the product itself become inhibitor for the enzyme that involves in the reaction. Okay. Okay, so let's look at the first one. Okay, so how enzyme activity are regulated. The first one is allosteric regulation. So if you look at this title, subtopic, it involves the word allosteric. Allosteric, it involves allosteric site, which is the site other than the active site where inhibitor or activator will bind. Okay, so that molecule that binds to the allosteric site, we call it as regulatory molecule. Okay, regulatory molecule can be activator, it can be inhibitor. Okay, so the regulatory molecule will only bind to the allosteric site, but the binding of the regulatory molecule, either activator or inhibitor, will bind just for a short while. Okay, briefly and reversibly. So the regulatory molecule can be removed. So the effects of binding of that regulatory molecule to that enzyme is just temporary. Okay. So a site, so allosteric site is, we know that it is other site than active site. Okay, active site only for substrate to bind, but the regulatory molecule binds to the allosteric site. So for example, here we have enzyme. So this is the active site where substrate binds. Okay, and then it has another site which is the allosteric site. So allosteric site we have, for example, other molecule can bind, such as inhibitor or activator. We call it as regulatory molecule. Okay. So the binding of this regulatory molecule causes the enzyme to change shape. Okay, change shape. Change shape, so when the enzyme changes shape, it also changes the shape of the active site. Okay, so if the active site change, it depends on the type of molecule that bind. So if activator binds to the allosteric site, it causes the active site to attain the active form so that substrate can bind. But if the inhibitor binds to the allosteric site, it causes the enzyme to change shape. Active site also changes shape that prevents the substrate from binding to the active site. So it depends on the type of regulatory molecule. Okay. So changes the shape of the active site either in the active or inactive form. Okay. So and then alters the activity and function of the enzyme. So for the allosteric regulation, there's two types lah tadi. So either allosteric activation by the binding of activator, or allosteric inhibition by the binding of inhibitor to the allosteric site. Okay. So if activator binds, it causes the enzyme to attain its active form. Yeah, so the enzyme changes shape to the active form. So the active site changes shape so that substrate can bind. But if the inhibitor binds to the allosteric site, it causes the enzyme to change shape so that the active site changes shape so that substrate cannot bind. So it changes into the inactive form. Okay. So that is allosteric regulation. So the one that you have to take note is this: regulatory molecule can be activator or inhibitor binding to the allosteric site, so causing the enzyme to be either activated or inactive. Okay. So next, okay. So here, okay, as you can see, it says here allosteric activators and also inhibitors. Okay. So for this one, it involves enzyme that has several subunits. Okay, so in this case, it has four subunits: one, two, three, four. Okay, so when enzymes, it has several subunits. We call it as allosteric enzymes. Okay, allosteric enzyme. Enzyme that has several subunits that has also allosteric site for the regulatory molecule to bind. But for this type of enzymes, the structure of the molecule is not stable. It is not stable. So the molecule will the enzyme molecule will keep on changing its shape. Active, inactive, active, inactive, active, inactive. Dia tak stable. Kan, dia bertukar-tukar bentuk, right? So in order to stabilize the enzyme structure, so it needs the regulatory molecule. Okay, it needs the regulatory molecule. So for example, here when the enzyme is keep on changing active form, inactive, active form, how to stabilize the enzyme? If if activator binds to one of the allosteric site, okay, activator only one, activator binds to one of the allosteric site, what happen is that activator binds to one of the allosteric site, it causes all the subunit of the enzyme to be in the stabilized shape, which is the active form. Okay, active form. So it requires only one activator to bind to one of the allosteric site to cause or induce all the subunits of the enzyme to be activated. Okay, to be activated. So these are the allosteric site, allosteric site, allosteric site, allosteric site. So dalam keadaan yang enzim tu berubah bentuk, aktif tak aktif, one activator binds to one of the allosteric site, causes all the subunit to lock into the active form. Macam tu, okay. But if inhibitor binds to one of the allosteric site, kan, so it causes all the subunit to be locked in the inactive form, stabilize the inactive form of all the subunit, and reaction cannot occur. So as you can see here, it says here a shape change in one subunit because one regulatory molecule, either activator or inhibitor, binds to the allosteric site. So the change of shape will be transmitted to all other subunits. So if activator stabilize all the subunit in the active form, if inhibitor stabilizes all the subunit into the inactive form, only requires one regulatory molecule to bind to one of that allosteric sites. Okay, so that is allosteric regulation involving allosteric enzymes, either activator or inhibitor binds to stabilize the enzyme molecule. Okay. If you have question, you can ask. Okay. Okay, so next, the second type of regulating enzyme activity is through the mechanism or process we call it as cooperativity. Okay, again, it involves allosteric enzymes, enzyme that has several subunits. Okay, given here, it has 4 subunits. Okay, so we know that for the allosteric enzyme, it is in a condition that it is not stabilized. It keep on changing shape. Active to active, active to active, active inactive, active inactive. So for cooperativity, so to activate the enzyme, to activate all the subunits of the enzyme, it requires one substrate to bind to one of the active site to cause all the subunit to be activated. That is cooperativity. So dia beza dengan allosteric regulation tadi. Okay. Allosteric regulation is through the regulatory molecule binding to the allosteric site. But for cooperativity, the word that you want is that substrate. One substrate binding to one of the active site, causing all the subunit to be locked in the inactive form. In the active form. You can say form, or you can say conformation, or you can also use the word shape, active form, active conformation. Okay. So causes once one of the substrate bind to one of the active site, causes the second and subsequent substrate to bind more readily, as all the subunit is in the active form. So what is the significance of this cooperativity? What is the importance? What is the benefit of cooperativity? It increases the rate of reactions. Okay, so as the enzyme has many subunit, right? So if the enzyme has many subunit, it can catalyze more. It can bind to more substrate, catalyze more reaction. So this amplifies the enzyme activity, producing more product, increases the rate of reaction rapidly. Okay, amplifies the activity of the enzyme. Okay, because the enzyme has many subunit, many active sites. Okay, so this amplifies the rate of enzyme reaction, enzyme activity. Ingat so cooperativity, substrate. One substrate to bind to one of the active site to lock all the subunit in the active form. So that is the keyword that you need to know. Okay, so next is the feedback inhibition. Okay, so feedback inhibition, activity of an enzyme near the beginning of a metabolic pathway is inhibited by the end product. Okay, so you have the initial substrate, okay, binding to the first enzyme. Okay, dalam sel kita ni, dalam sel dalam badan kita ni, reaction dia bukan sekadar okay, kita ada substrate, enzyme terus hasilkan produk. We have this initial substrate binding to the first enzyme and then produces intermediate product and then catalyzed by several other enzymes until you produce the end product. Okay. Once the end product has been produced, okay, so there will be an accumulation of the end product. So you do not want the cell to over producing that end product. Okay. So at one point, you have to stop the production of the end product for a while to let the cell to use that end product. Okay, so this end product that is produced, can so it becomes the inhibitor for the first enzyme of the pathway. First enzyme. So the end product becomes the inhibitor for the first enzyme. Okay, so the end product become the inhibitor for the first enzyme of the pathway by binding to the allosteric site of that enzyme. Okay, so this is called as allosteric inhibition. So the increasing concentration of end products serve as a signal for the enzyme to slow down and stop functioning. Okay, stop functioning. So you do not want the cell to produce end product excessively. Again, so in the increasing concentration of end product serve as a signal for the enzyme to slow down and eventually stop functioning. Stop functioning. So what is the significant? What is the benefit or what is the importance of this feedback inhibition? Prevents the cell from wasting chemical resources. Faham? Okay, so kalau sel tu dah cukup, contohnya dah cukup menghasilkan ATP contohnya kan. So ATP awak you just apa you your cell has produced enough ATP but you are not moving. You just lay down after you eat and then you lay down and then you go to sleep. So that process of producing the ATP molecule is produced, but your cell is not using that ATP because you are not moving. For example, you are not using that energy. Contohnya, kan? So the cell will stop for a while the production of ATP molecule. Okay, so the cell will not produce ATP continuously without the cell use that energy for the activity within the cell. For example, movement, muscle contraction, to move substance against concentration gradient. There's so many usage of ATP molecule as we know. Okay, so that is the benefit of feedback inhibition. It prevents cell from wasting chemical resources. Isma, do you have any question?
Madam,
Yeah.
contoh first enzyme apa?
Kita tengok je lagi ada contoh dia.
Oh, okay.
Ada reaction dia. Okay. Alright. So this is just the the concept behind feedback inhibition. So we are going to look at example. Okay. So feedback inhibition, such as for example the synthesis of isoleucine. Isoleucine synthesis. Isoleucine is a type of amino acids. Okay. So this pathway, if I'm not mistaken, it involves the reaction that occurs in bacterial cell. If I'm not mistaken, you can check. Okay, isoleucine synthesis. Okay, so for this one, it says here synthesis of isoleucine from threonine involve 5 steps. Okay, so here we have here the initial substrate. The initial substrate is threonine. Threonine is the initial substrate binding to the first enzyme. In this case, the first enzyme is threonine deaminase. Okay, so when threonine binds to the enzyme, which is the first enzyme, threonine deaminase, produces the first intermediate product, intermediate A. And then to produce intermediate B, catalyzed by the next enzyme. And then produces intermediate B and then catalyzes by the next enzyme, produces intermediate C, catalyzed by the next enzyme, produces intermediate D and then catalyzed by the last enzyme, and then produces the end product. The end product that the cell produces, that the cell wants, is isoleucine. But when the cell has produced enough isoleucine, there's already so many isoleucine in the cell. So the cell will stop the production of the product by the end product becoming the inhibitor for the first enzyme. Okay, for the first enzyme, which is threonine deaminase. As you can see, threonine deaminase, it has allosteric site. So the end product, which is isoleucine, binds to the allosteric site of the enzyme, of the first enzyme, causing the enzyme to change shape. Active site changes shape and substrate cannot bind. So this stops the reaction for a while, only temporary. The effect is temporary. Okay, temporary. So it is reversible. Okay, reversible. So so this allows the cell to use up the isoleucine, and then isoleucine concentration will be reduced after the cell detects the isoleucine inside has been reduced. Then the isoleucine will be removed from the first enzyme, and then the enzyme will transform back to its original conformation and then catalyze the reaction again to produce more isoleucine. Okay, so that is the concept behind feedback inhibition. It says here, okay, so the synthesis of isoleucine from threonine, which is the initial substrate, involve 5 steps, 5 enzymes, and then each step is catalyzed by different enzyme. And then we have the enzyme. The first enzyme is threonine deaminase. catalyzes the first reaction until at the end produces enough isoleucine. Isoleucine concentration has increased or accumulates and then becomes the inhibitor for the first enzyme by binding to the allosteric site and inhibit the activity of enzyme threonine deaminase. Okay, so the importance of feedback inhibition macam tadilah prevents the cell from synthesizing the product which is in this case isoleucine then is necessary. Macam tu lah kan. So ini contoh untuk isoleucine. Sama juga kalau macam contoh the in the production of ATP kan. So it is the same kan. So it allows the cell to use up the product first and then after the cell detects or the product has been used up and then the product concentration has been reduced, then only the reaction can occur again. Macam tu lah. Okay. Okay, Isma boleh?
Boleh madam, thank you.
Okay, so yang lain okay Alright. If you have question, you can ask. Okay, so because it is 4:49, I guess you want to continue or stop?
Stop.
No, stop. Stop, cepat je stop. Okay, so we stop dulu. Do you have any question regarding today's lecture? Ada soalan?
No.
Okay, so just to recap, to wrap up, the one that we have looked at during this lecture is that you have to know, okay, there's condition within cell that regulates enzyme activity. Okay, first is the presence of regulatory molecule, either activator or inhibitor. And then there's the process, we call it as cooperativity, which is substrate binding to the enzyme. One of the enzymes active site to activate all the active site and increases rate of reaction rapidly, amplifies the rate of reaction. And then we have feedback inhibition, where the end products become the inhibitor, allosteric inhibitor for the first enzyme of the pathway that allows the cell to use up the end product first and prevents chemical wastage. Okay. So yang tu tolong buat revision balik. So ada soalan? Do you have any questions? If you don't have any question, I guess we can end our session for today. Okay. So thank you, class.
Thank you madam.
Okay.
Thank you, madam.
Thank you, madam.
Okay.
Thank you, madam.
Bye bye.